is leading these other consortium. As you can tell, we're consortium people. We just join consortium, trying to do interesting work with other people. So there is this Zoonovia consortium, and they published a paper by sequencing 240 placental mammals, the genomes of 240 placental mammals in this paper. And Eleanor listened to the presentation of a graduate student of mine, Greg Andrews, and said, why don't you join force with us and take a look at the evolutionary conservation of these regulatory elements. So, Kylie Fan is another PhD student who has now defended as well, and Henry, as you can see why I brought Henry here. Henry is part of the mix of all the things we're doing here. So the three of them teamed up to look at the evolutionary conservation of these CCREs in mammals. So we say, okay, let's make the same plot that I showed you earlier, average vital P score. So these dashed lines are the lines that I showed you earlier across 100 vertebrates. And we make the same plot for 241 mammals. So 240 and 241, the one is human. Actually, no, that's actually not true. So a side story, just to keep you alert, is Eleanor is actually a dog person. She studies a lot of dogs, using dogs as a model organism for human diseases. So the difference between 241 and 240 is 241 is the number of genomes, 240 is the number of species. And she snuck in 2 genomes of dogs. One is purebred, and the other one is like mixed bred because of her background. So we end up, sometimes you will see 241. It's because there are 2 dog genomes in there.
Is there a control curve there? Like the
oh yeah, the random curve I failed to include. You know, for reviewers, I actually had to add back the gray line. I do have the gray line.
Yeah, I reviewed that paper.
Yes, absolutely, Paul. So the gray line for 241 mammals increased a little bit. It moved from like here to here. So it elevated a little bit, but not enough to account for the huge increase in the conservation. So we actually did more detailed work by mapping things into this simplex. So we have 240 species, right, keep in mind. So we looked across the species and see across these CCREs. I failed to alert you. These CCREs are like the size of 1 nucleosome. So they're like between 150 and 350 nucleotides long. So roughly the size of 1 nucleosome. So we can look across the length of the CCRE and say, out of the 240 species, or 241 genomes, how many of them can align more than 90% of the positions in a CCRE, or how many of them can align less than 10% of the positions in a CCRE. If you calculate those 2 numbers, and each CCRE will fall into this triangle. And just by definition, they have to be on this triangle, and this corner is belongs to the CCREs that are highly conserved across the species, and that corner belongs to the CCREs that are least conserved across the species. Okay, and this is another corner that lights up, and you can see this kind of structure is partly determined by the conservation of the CCREs, and partly determined by which kind of species we have across the 240. So in comparison, this is a random region. You can see this region is more populated for CCREs than randomly chosen genomic regions of matching sizes. And then this region is less populated for CCREs than random regions, to indicate that CCREs are on average more highly conserved. That's what I have told you already. But the interesting thing is 50% of the CCREs are highly conserved across 240% of mammals. I have a comparison between, this is the same plot as in the previous slide. In comparison, if you make the same plot for the 100 vertebrate genomes I mentioned earlier, and you get a very different picture. This cluster of CCREs that are highly conserved across percent of mammals moved to this corner. And it's a complicated picture. I don't want you to get distracted, and this is the random comparison, but I just want to mention that the conclusion we get from this comparison is less than 5% of the CCREs are conserved beyond the mammals. And that's why if we use the mammalian genomes, it gives us the highest sensitivity of quantifying evolutionary conservation. Okay, Martin looks quizzical. Okay, good. Okay, so we can also not only just use the percentage of alignment, we can actually take the alignment and physically make a matrix of the alignment. So for each CCRE, and you can tell what percentage of the position is aligned in each of the 240 mammals, right? And that's a big matrix. And if you use a dimension reduction technique, such as UMAP, on that matrix, for the entire 1 million of CCREs, that's what you get. It looks like a flag. I was very intrigued by this highly organized structure of our regulatory elements. So group 1 is the group that's highly conserved. Group 2 is the one at the lower left corner. Those ones, we call them actively evolving. They are kind of a mixed bag. And then group 3 are the least conserved. So there is a continuum from the highly conserved to the least conserved. And the most intriguing thing is the least conserved ones just exploded into little discrete clusters, which was quite intriguing to us. And I added these two plots to indicate this green group here, group 2, actively evolving, that really depends on the cutoff of how many genomes need to be aligned for whatever threshold of the length of the CCRE. And you can see that they are partially conserved, and that's why we call them actively evolving. So the same UMAP, now you can color it every each way you like. For example, this is the same UMAP, but colored by the Philo-P score. And the highest Philo-P score, not surprisingly, corresponds to the tip of the UMAP. And then if you color it by distance to transcription start site, it also shows up these ridges of the map that corresponds to regions, the CCREs that are near TSS. Everything makes sense. And if you overlap with the transposable elements, so TEs, the blue ones that overlap transposable elements, those are from the end of the map that are least conserved. And then the most conserved end of the map do not overlap with the transposable element. So that was the evolutionary conservation. I'm interspersing 2 lines of work. So we continue into ENCODE phase 4, and now we increase the number of our CCREs from like 1 million to like 2.3 million now. We actually, the final version 4 is the one that's being worked on by Joe Moore. And 2.3 million is what we have now. And then we have been refining the categories, in case you're wondering, the ones we have. I'm not going to go into details. So that all these presumably will show up in a companion paper we are working on right now. So just a little bit of detour as to what Henry will talk about. We actually spend a lot of effort to make our work accessible. So Henry's going to talk about screen. I'm not going to go into details. So Michael and Henry built this visualization tool that's part of the encodeproject.org. If you go to the ENCODE portal, and you can access this tool from there, they'll bring you to the site, and Henry will talk about more details. So yeah, there is a movie, so I am not going to go into details. So basically, if you go into the site, you can search, you can browse, you do all kinds of things, answer individual questions about CCREs individually or as a group, and also link CCREs with possibly their target genes using other kinds of experiments. I mentioned high-C, chromatin activity, SharePad. QTL, expression quantitative trait loci. These are additional experiments. These are single cell, kind of like QTL. So we have all these data basically in screen. It's like a repository of annotations around CCREs. And I'm going to skip this. And also, I mentioned a lot of ChIP-seq experiments for transcription factors. Henry Bragg and then Nishi is a bioinformatician in my lab. The three of them put together this factor book resource, which is the equivalent resource for transcription factors, similar to screen for CCREs. And I have a summary of a factor book with thousands of ChIP-seq experiments in ENCODE, and human and mouse. So for each ChIP-seq experiments, we have a set of peaks, and then we annotate it with a pipeline that goes through, identifies sequence motifs of these transcription factor binding, chromatin profiles around the transcription factor binding sites, and then also try to connect these sites with pathophysiological context that might explain the impact binding by transcription factor. So it's published, and factorbook.org is the site if you want to browse around. So the key thing is we can identify the peaks and the high confidence motif sites for whatever number of experiments that ENCODE has performed, which is thousands of experiments covering hundreds of human and mouse transcription factors. So, for each of transcription factor, we can find the sequence motifs for them. So here is a paper that in NAR that's Henry's work. So you can basically perform a similar low dimension reduction visualization of these motifs, and you can see that different transcription families bind to clusters of motifs that are similar. And from this annotation effort, we identify novel motifs that haven't been annotated before. And these motifs, if you want to see how confident we are, you can use these peak centrality, evolutionary conservation, and DNA's footprint, all these auxiliary experimental data to show, to reveal whether or not the motif is likely to be a real one. So this is like a parallel effort that we have been doing, and I'm introducing them right now because I want to go back to the evolutionary conservation angle for both CCREs and transcription factor binding sites. So just one more explanation. So all these transcription factor binding sites, they are much smaller than the CCREs. So remember, CCREs are like 200 base pairs long, and TF binding sites are anywhere between 6 to 20 base pairs long. So each CCRE may contain like 5 to 10, 20 sometimes transcription factor binding sites. And I remember, Martin, we had quite a few papers about clusters of transcription factor binding sites. And it's the same idea, but now it's reframed in the context of individual CCREs. So, so if you still remember the UMAP for CCREs, we can make exactly the same UMAP for the binding sites of each transcription factor. So we have a lot of UMAPs. Okay, so here is an example of these hepatocyte-specific liver factor, HNF4A. So roughly 500,000 such sites in the genome, and we can find out whether or not the site is conserved across these 240 mammals, and make that huge matrix and dimension reduction, and it's similar. So group 1 is red, is the most conserved. Group 2 is green here. We have a lot fewer group 2 actually. And then group 3 becomes much more discrete. So we're very intrigued by these individual clusters of group 3 HNF4A binding sites. So what are they? Okay, So we can look at each group in the context of the evolutionary tree. So we have a tree of 240 mammals. This is just the tip of the tree. Human is not included in this tree because all these sites started in humans. That's why they are in humans by default. You can see that A is this blob here, and these are 1,500 sites that are in great apes, including human, but not in other species. And cluster B is here, are the sites that are in great apes and old world monkeys, and not in other species. I'm not including the ones down below because they're all blue, and I don't show them. And then different clusters of sites just indicate different distributions across the species. Is there any question? No? Okay, good. So, and also, all these sites are derived from transposable elements. And yeah, some of the earlier talks described transposable elements. So if you look at the transposable elements in the human genome, there are line 1, line 2, Alu is the sign element, Mir is an ancient sign-like element, Mir. This is ancient, and LTR, long terminal repeat, DNA element. So these are different kinds of elements. The interesting thing is you don't need to remember the details of them, but the one thing that would be useful is if you could remember line 1 is the youngest family of elements. Alu is relatively young. Mir is very old.